Okay, so hi, my name is Angel Ko, and I will be presenting on my research that I have been conducting with Liu Jin, a PhD candidate. Today, I will speak about how we utilized optogenics to examine the functionality of tissue-engineered nerves with a minimally invasive method. Only some neurons developed from engineered nerve tissue models are functional and considered desirable. Therefore, it is necessary to remove the undesirable non-responding neurons to obtain the most reliable results. Currently, one of the methods used to distinguish between these neurons involves the use of microelectrodes. This is an invasive technique that penetrates the cell body to test whether depolarization occurs for the induction of action potentials. Naturally, this will render some of the desirable cells non-functional, as it may disrupt the cell structure. Since this is difficult to execute in a large scale, it is time-consuming to test all of the neurons in a sample. And the excessive contact with the neurons also introduces the increased likelihood of errors. The ultimate aim of this research is to develop a quick and minimally invasive method involving the use of optogenetic regulation of cells that can detect the neuronal activity in order to evaluate the functionality of our engineered nerve models. I will use a viral vector to insert a gene for the production of ion transport channel protein, channel rhodopsin 2. After stimulated by blue light, the protein will undergo a physical conformation and depolarize. So for our background, we will utilize lentivirus as the viral vector to insert the gene. To increase the safety of handling the virus in the lab environment, the components for the virus production are split amongst 3 plasmids: the lentiviral transfer plasmid encoding the gene of interest, the packaging plasmid, and the envelope plasmid. These 3 will co-transfect our 293 T cells to produce the lentivirus containing the gene for channel rhodopsin 2. Lentiviruses are capable of infecting non-dividing and actively dividing cell types, which is a greater variety of cells than retroviruses can. It is also a more stable alternative to other viral vectors such as adeno-associated viruses. The objective of my research is to manipulate the viral vector lentivirus to insert the gene of interest into my target cells. The gene of interest codes for the blue light sensitive channel protein channel rhodopsin 2. After the application of blue light at 470 nanometers wavelength, the CHR2 will undergo a conformational change to allow a flow of positively charged ions into the cytoplasm, causing depolarization. This will result in the propagation of an action potential. The lentivirus will insert the gene into induced pluripotent stem cells and neurostem cells. After, I will differentiate the successfully transduced cells. These cells will be cultured on a specially designed plate with a multi-electrode array at the bottom surface. These electrodes will record and process the biological signals. Any neuronal action potential deciphered from those signals will be strong evidence to prove the usefulness of a new approach of activating neuronal cells by light stimulation. This technology is expected to carry out the experiments free of cell damage and yield substantially more accurate data as compared to the conventional methods. So for my materials, the 3 plasmids that we used are Plex 307, which contains CHR2, our gene of interest, PSPAX2, and PM2GG. These cells we transfected the plasmids into are the 293 T cells, and we have transduced the viruses into the neural stem cells. So the Plex plasmid is a lentiviral transfer plasmid that encodes the gene of interest, channel rhodopsin 2. In addition, it is tagged with M-cherry fluorescence, which will allow us to visualize which cells are transduced successfully. The ampicillin and pyrimycin resistance is to help us further purify our samples and remove extraneous substances. PMD2G is our envelope plasmid and also contains ampicillin resistance. PSPAX2 is the packaging plasmid component, which also has ampicillin resistance. So part 1 of our project is the manipulation of the viral vector lentivirus into insert the gene of interest into our target cells. Our method to accomplish part 1 involves 3 steps. The first part is the bacterial culturing and plasmid purification. The 3 plasmids we are using have to be cultured and expanded before purified to verify their DNA sequential identities. After confirming the plasmid identities, we will begin 293 T cell transfection. 293 T cells are highly transfectable derivatives of human embryonic kidney 293 cells. These cells are used as the vessel to produce copies of the lentivirus, which will be secreted into the extracellular matrix. The last step involves the transduction of these viruses into our target cells, the iPSCs and NSCs. So for our bacteria culture, we streaked the agar plates in 4 quadrants. After incubating overnight, we used the single colonies to further inoculate in the LB liquid broth to create a starter culture. After incubating the starter culture for 8 hours, we diluted the sample in a flask with ampicillin and let shake overnight. After the bacteria have grown, we utilize the MIDI prep kit to isolate the plasmid. The kit provides the buffers to allow us to lyse the bacteria, which allows the plasmid to enter the supernatant. After centrifugation, the supernatant is transferred to the tip to be filtered. The plasmid binds to the resin in the tip and is washed by various buffers before elution. After elution, we utilize isopropyl to precipitate the DNA. In the image, you can see a difference in the color of the CHR2 and PSPAX2 DNA plasmid solutions. After extracting the plasmid DNA, we ran gel electrophoresis on the samples and sent the samples to be sequenced. We received positive results on both tests, so we proceeded with transfection. Transfection is the process of artificially introducing nucleic acids into cells. Electrophoresis is a transfection method that uses an electrical pulse to create temporary pores in the cell membrane to allow the plasmid to enter. We tested 2 different conditions so that we may understand which condition may be more beneficial in receiving successfully transfected cells. To determine whether the plasmids have successfully transfected into the cells, we used an eclipse microscope to verify that the M-cherry fluorescence is present inside the cells. We found that the cells were successfully transfected, so we proceeded to harvest the viruses. After the cells began expressing the virus, they excreted into the extracellular matrix. We used PEG to concentrate and precipitate the lentivirus, then resuspended the virus in the resuspension solution provided in the kit. So this is our picture of day 1 cells. On day 1 of our transfection, we seeded the cells in 2 different confluency levels. Our first expel plate was about 28 plus or minus 7% confluent, while our second plate was about 52 plus or minus 6% confluent. So we cultured the neural stem cells with varying concentrations of lentivirus, which were harvested from 293 T cells transfected under different electroporation conditions. Polybrine is a polycation that neutralizes charge interactions to increase the binding between the vector and the cellular membrane. While this is beneficial to our transduction, it is also harmful to the cells. So we are testing 2 concentrations of polybrine to determine the optimal condition. On day 2, before we changed the media for the cells, we were able to see the M-cherry fluorescence. This means that the cells are producing the protein channel rhodopsin 2. In the control, you can see that there is no presence of lentivirus, so there is no fluorescence. There is no protein production either. In the 100 volts per 5 milliseconds condition, you can see the red lights within the cells. In the 125 volts per 2.5 milliseconds, there's about the same concentration of fluorescent cells. On the 4th day before we harvested the virus, it is noticeable that the concentration of fluorescent cells have grown. The cells in the 100 volts per 5 millisecond conditions are brighter as it produces more protein over time. However, there's a higher concentration of fluorescent cells in the 125 volts condition. Therefore, we can conclude that the second electroporation condition is more suitable for transducing cells. On day 8, after we rinsed the cells with pyrimycin, we are left with dense colonies, which tend to be transduced cells. There are higher concentrations of fluorescent cells in the condition with the electroporation and polybrine. And for part 2, since we have just recently successfully transduced our cells, we have yet to begin the second part, which is differentiating the transduced cells into neurons. This is an image of a differentiated neurostem cell. The red stains show the actin, which is the cytoskeleton of the structure. The blue is staining the nucleus to show where the cell body is. And the green is the neuronal marker to show where the proteins are present. I would like to thank my principal investigator, Dr. Jin Nam, for providing me with the opportunity to work on this project and for guiding me through it. I'd also like to thank Lu Jin, my partner, that I have been working on this project with. Thank you.
Great. Thank you so much, Angel. You can go ahead and stop your recording now.